As we begin our study of genetics, let's go back and review some things that you've learned before about Mendel's laws, about Mendel's experiments and Mendel's laws. So remember that although it's easy to think about inheritance as being a blending of traits from the two parents, sort of like green and yellow, blue and yellow paint blend to make green paint, the idea that we use today to talk about inheritance is something called the particulate hypothesis, and that means that we've got heritable units that we call genes, and this is much more able to explain some of the phenomena that we see in inheritance, like the reappearance of traits after being absent for several generations. And we owe our understanding of the way this works to Gregor Mendel, who experimented with garden peas. This is a picture showing some of the monks at the monastery where Mendel was a monk. This is Mendel right here, looking at his pea plants, and everybody's kind of looking at him like, what are you looking at? But anyway, this just kind of shows you as a reminder that Mendel was a monk. He worked in a monastery. He did study biology in college. Or he did go to university and study, but he used his understanding of scientific principles to do the experimentation that he did over a number of years at the monastery. He used the scientific method, as we call it today, to identify the two main laws of inheritance, and he did this by breeding garden peas, and his experiments were very carefully planned. He counted lots and lots of peas over the course of time. He chose peas. I'm not really sure why he chose peas, but he was pretty fortuitous in his choice of peas. There are some big advantages here. For one thing, there are different varieties that have different kinds of characteristics, like color of the flower or the length of the stem or things like that. And those are called traits. Those are the variants, the differences between one color of flower and another. We can control the mating of peas. Peas they generally self-pollinate, but it's pretty easy to keep them from pollinating themselves. Every flower on the plants has both male and female parts, the stamens and the carpel, and you can easily cross-pollinate one plant with the other with putting pollen from one plant onto the carpel of the other one. So it's pretty easy to handle. This shows you the basic technique. Here's a flower of a pea plant, and you can easily cut off the stamens here and leave behind the carpel, and then use some kind of mechanism. He used a paintbrush or something similar to that to transfer pollen from the stamens of a different flower plant onto the carpel that was unable to be fertilized by its own stamens. And then this grows into the peas, the fruit there, which is the pea pod containing the peas, and then you can plant those peas and see what happens. And so one of the original experiments he did, of course, was to cross purple flowers with white flowers, and he found that the first generation of offspring actually all had purple flowers. So he looked at specific traits that only had two very distinct alternating forms, okay? And he started off with varieties that were true breeding. In other words, when they were allowed to self-pollinate, they produced offspring all like themselves. So what he would do is make these two different varieties. This is called hybridization in the world of botany or horticulture. The true breeding parents were called the P generation, the parent generation, and then the hybrids of the P generation are called the F1 generation. And then when they self-pollinate or cross-pollinate with other F1s, then we produce the F2. The F stands for filial, which is the offspring, our brothers and sisters, basically the children. So the F1 would be the first generation after the parents. F2 would be the second generation after the parents. So when he crossed these true breeding flowered plants, white and purple flowered plants, then he found, as we saw in the previous picture, that all of the hybrids from the F1 generation were purple. When he crossed those with each other, allowed them to self-pollinate. A lot of the next generation, the F2 generation, had purple flowers, but some of them had white flowers. So what he did in keeping track of this, he did lots of meticulous accounting and counting of peas and flowers and so forth. And so what he found over time was that there was a ratio of about 3 to 1 purple to white flowers in that second generation. So here we have our true breeding parents, the P generation, and our F1 generation, the hybrids that had purple flowers. They self-pollinated or cross-pollinated with each other. And the F2 generation had 705 purple flower plants and 224 white flower plants, which is about a 3-to-1 ratio in terms of the numbers there. He figured out that what it was, whatever it was that was making the flower purple, was affecting the flower color in those hybrids. And so he called that trait, the purple trait, a dominant trait, and the white flower would be a recessive one. And then it's not that it was destroyed, because it did reappear in the second generation, so it wasn't totally, it was covered up basically, or prevented from being expressed, as we would say these days, but it still could show up in another generation. He saw 6 other plant characteristics, 7 total characteristics that he looked at, each one represented by 2 traits, a dominant and a recessive, and he called these heritable factors. We now call them genes, but he called them heritable factors. Now remember, when Mendel was doing his experimentation, he didn't know about chromosomes, he didn't know about meiosis, he didn't know about any of those kind of things, gametes and somatic cells and so forth. He did not know about that. So he was just going strictly on the numbers of what happened in the traits that showed up in the offspring, not really understanding the mechanism. These are the 7 characteristics that he looked at: flower color, the position of the flower, whether on the ends of the buds or on the branches or on the ends of the plant, the color of the seed, either yellow or green, the shape of the seed, either round or wrinkled, the pod shape, either inflated or constricted, and the color of the pod, and the length of the stem. So all of these different things he looked at, and he got the same sorts of results every time he crossed them. So it was almost exactly 3-to-1 every time that he did the same kind of hybridization, F1 and F2 experimentation. So he came up with a hypothesis to explain this, and there are 4 concepts that are involved in this. 3 we can relate these to what we now know about genes and chromosomes, but he didn't know about genes and chromosomes, so he couldn't make that leap that we can make. First of all, there are alternative versions of genes that account for these differences in the characteristics. For example, the flower color. There are 2 versions of it, purple and white. These are nowadays we call these alleles, the alternative versions of the gene. And then we also know now that each gene resides at a specific location or a locus on a specific chromosome. So it's not just random. There's definitely an order in which they occur. Here we have 2 chromosomes, a pair of homologous chromosomes, one from dad, one from mom, basically. And we've got an allele. The locus for the gene is in the same location on those homologous chromosomes, but the alleles can be different. So here's a purple flower allele and a white flower allele. Secondly, each characteristic, for every characteristic, the offspring inherits one copy of one allele from each parent. And he didn't know anything about chromosomes, so he couldn't say that one chromosome comes from mom, one comes from dad, like we say today. The 2 alleles can be the same, and that would produce a true breeding plant. And if they're different, then you would end up with a hybrid. The third statement would be that if those 2 alleles are different, then one of them is going to determine what the organism or the offspring looks like, and the other one is not going to have an effect on the parents. Nowadays, we know that a lot of genes don't particularly code for something in the appearance, but we'll still use that as a convenient way to describe it. So in the case of the flowers, the allele for purple flowers is dominant to the one for white flowers, and so it's the one that shows up rather than the white flower. The fourth statement, this is now called the law of segregation, says that these 2 alleles for this gene are going to separate during the formation of gametes and end up in different gametes. And so an egg or a sperm is going to get only one of the 2 alleles that are there for that particular trait. And nowadays, again, Mendel didn't know about chromosomes. He just knew that one copy of each inheritable characteristic came from each parent. And nowadays we can talk about the homologous chromosomes and the distribution or separation of them in a meiosis into different gametes. And that explains how you end up with only one copy in each gamete. This is what accounted for this 3-to-1 ratio that he had that he saw in the F2 generation. You can look at a Punnett square, of course, to see the different combinations, different possible combinations. And nowadays, by convention, we show the dominant character as a capital letter and a lowercase letter representing the recessive character, the recessive allele. When we do our cross showing a Punnett square. One thing I always caution my students to remember that a Punnett square is a probability. It's showing you the probability of what could happen, not exactly what will happen. And the way I usually refer to this in class is it's a probability, not a promise. So just because you have possible 4 different outcomes doesn't mean that you'll get one of each. And each, one-fourth of each type in each reproductive cycle. It's a probability, not a promise. So here we have our parent generation again, showing that we have the purple flowers and white flowers. Now we can show the letters that stand for those. So these true breeding parents, purple flower would be 2 dominant alleles, and the true breeding white flowers would show 2 recessive alleles. Homozygous dominant, homozygous recessive. When we produce gametes, we're only going to get 1 copy of each one. So the one gamete would have the capital P for the purple, and one would have the lowercase p for the white. When they're combined together in the F1 generation, then you have still purple flowers because you still have one of those dominant alleles. But notice that you've got 2 different alleles there. You've got a purple and a white, or a dominant and a recessive together in this heterozygote. And then finally, in the F2 generation, then what you end up with is a 3-to-1 ratio of purple to white. You can see that the sperm from the F1 plant would produce a big P and a little p, 2 different types of gametes for purple for the flower color characteristic, and the eggs would also have only 2 different kinds of alleles for the flower color. And so you end up, when you do the Punnett square to show the cross, that 3 of those are going to be purple, either homozygous dominant or heterozygous, and then 1 would be homozygous recessive, which would be the white. Now, there are some vocabulary words, and I've used a few of these, so you should make sure that you understand how these fit together. When you have 2 identical alleles for this characteristic, that's called homozygous, homo meaning the same, zygote referring to the fertilized egg. When you have 2 different alleles, that's called heterozygous. Hetero means different. And the thing that you need to remember about heterozygotes is that they are not true breeding. The homozygotes will be true breeding for that characteristic. The heterozygotes will not be. The traits aren't always going to show the genetic composition. So if you see a purple flowered plant, you can say, well, it's either homozygous dominant or heterozygous. We don't know which one. The homozygous recessive one, the white one, you'll always know what that one is. And so we have to look at the difference between the phenotype, the physical appearance, or the expression of the trait, and the genotype, which is the genetic makeup. And so even though purple flowers have the same phenotype, the same appearance, they can have different genotypes, either the homozygous dominant or the heterozygous. This just shows you an example of what that looks like. So you see that these, although the phenotypes are all purple for these, 1/4 would be homozygous dominant, and 2/4 or 1/2 would be heterozygous, and leaving the 1/4 that would be the homozygous recessive. In this case, the white flowers. Notice also we've got generally a ratio in the phenotypes and genotypes, and the ratios are a little bit different. Okay, when we look at the phenotype ratio, that's the appearance, then the ratio would be 3-to-1 basically. When we look at the genotype, then we're going to see a ratio of 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Again, probability, not promise. Now, when we want to figure out that dominant phenotype and what the genotype is, we have to have a way to do that. This is something called a test cross. Okay, and so when you do a test cross, you're going to breed that dominant phenotype with a homozygous recessive individual. And if you've got any offspring in that first generation that are showing the recessive characteristic, then that tells you that your dominant parent was heterozygous. If they're all the dominant phenotype, then chances are very high that that individual was homozygous dominant. So here we have our dominant phenotype. We don't know what the genotype would be, but we do know the genotype of the recessive. Okay, and so this would be your prediction. Okay, remember it's a prediction; it's a probability, not a promise. In the case of a homozygous dominant, you would expect all of the offspring to be purple. If it were homozygous recessive, then in this case it would be half purple and half white. So if you had any white ones at all, you would know that there was a heterozygous individual for that dominant characteristic. The law of independent assortment is a second law that Mendel came up with. The first law was the law of segregation, and he was looking at one single characteristic. And we're producing an offspring there where we're looking at monohybrids. That's going to be heterozygous for one characteristic. And that's called a monohybrid cross. If we're looking at two different characteristics at the same time, that's when we're going to come up with our second law of inheritance, which is the law of independent assortment. So crossing two of these true breeding parents, okay, is going to produce dihybrids in the F1 generation. They're going to be heterozygous for both characters. So whatever the true breeding characteristics are, we're looking for two different characteristics here. And then when we cross the F1 dihybrids, then you can figure out whether those traits are packaged together or whether they are separated independently of each other. In the case of Mendel's traits, his definitely separated independently. We'll see later on that's not always the case. So here's our parent generation. We're looking this time at the seed, okay? The yellow seed, believe it or not, in peas is a dominant characteristic, even though it's less common. And the round shape is also the dominant characteristic, and the green and the wrinkled characteristics are recessive to the other traits. So here will be our gametes from the first one. The capital Y, capital R would be our two dominant alleles, and then the lowercase y and r would be the recessive alleles. So our F1 generation are all going to be round yellow, okay, but they're going to be hybrid for two different traits. So what would be the case? Well, half of your offspring, or half of your sperm cells, if you just had a dependent assortment, if they were dependent on each other, then that would mean that the yellow and the round were tied together. Okay, and so then half of your sperm cells would be capital Y, capital R, and the other would be lowercase, and the same thing with the eggs. And so in that case, you would expect to get a phenotypic ratio in your offspring of the F2 generation, 3:1 characteristic. But if you have independent assortment, if they're not tied together, then you have 4 different possible combinations of the alleles. Remember, when we're determining the allele combinations for the gametes in a dihybrid cross, we're going to use the FOIL technique that you learned in algebra long years ago, where you take the first one of each pair, the outside ones of each pair, the inside ones of each pair, and the last one of each pair to make your combinations. So we have 4 different possible combinations of alleles in the gametes, and we end up with this kind of ratio. In the phenotypes, we end up with a 9:3 to 3:1 ratio. 9 homozygous dominant for both, or 9 dominant for both, 3 dominant for one, recessive for the other, 3 recessive for the first and dominant for the second, and 1 that's homozygous recessive for both. And so what he got with his results were these numbers down here: 315, 108, 101, and 32. And if you work out the math, it works out pretty close to 9331, which would be our expected. So when Mendel looked at this cross, this dihybrid cross, he came up with this law of independent assortment, which states that a pair of alleles is going to separate independently of each other of each of other pair of alleles during gamete formation. So what this means is this is talking about the chromosomes rather than just the alleles. It's talking about the chromosomes. Okay, you're only going to, each chromosome is independent of each other chromosome. Now that's only really going to apply to when you're talking about genes on different non-homologous chromosomes, or ones that are very far apart, because crossing over can occur, and if they're far enough apart on the chromosome, then the crossover ratio, it makes them separate as if they were independent of each other, totally independent. If they're close together on the chromosome, they're generally going to be inherited together. Ones that are farther apart are generally going to be separated. And this turned out to be the case with Mendel's alleles. Of the 7 traits that he studied, 6 of them were on separate chromosomes. There was one pair, two different traits that were on one chromosome. Now that we've learned more about chromosomes, but they were so far apart from each other that they separated or segregated independently of each other. This concludes the basics of Mendel's laws.